if the histones were mutated and made rich in acidic amino acids such as aspartic acids and glutamic acid in place of basic amino acid such as lysine, will result in a lack of packaging of DNA and resulting in no chromatin formation. Now you have to say whether this statement is true or false. Now we see, if you look at the basic amino acids, lysine, arginine, these are positively charged amino acids and DNA is a negative charge. So these two gets attracted and therefore a proper packaging of DNA takes place. But according to this statement, if we replace this or if they are converted into an acidic amino acid, then they will have a negative charge and it will result in repulsion, or this DNA and the negative amino acid like glutamic acid will repel and therefore proper packaging will not take place. So this is a true statement. Second, a single base mutation in a gene may not always result in loss or gain of a function. So now if you look at the wobble effect, for example, we have a codon where GGU, GGA, GGC and GGG. Now if you look at these, first two nitrogenous bases are same and all these they code for a single amino acid called glycine. Now if you look at this, we can easily understand that it is the first two nitrogenous bases which are important and the third one will not have any impact on the coding of the amino acid. So even this statement is a true statement. By repeating Hershey and Chase experiment with 32, that is radioactivated phosphate, and 15 nitrogen instead of 35 sulfur, it will yield exactly the same result. First of all, this is not a radioisotope, it is a heavy isotope, whereas this is a radioisotope. Now if we replace this 35 sulfur with the nitrogen, now what happens is this nitrogen is a structural component of both amino acid. This is the component of amino acid and this is the component of nucleotide or DNA. So if we incorporate a heavy isotope, what will happen is it will insert or it will get inserted into both protein and DNA. So therefore the purpose of the experiment will not be the same. It will be unable to detect whether the genetic material is DNA or RNA. So this is a false statement because it is not going to result in exactly same result. Alternative splicing of exons may enable structural gene to code for several isoproteins. Now for example, alternative splicing results in different combination of exons. So E1, E2, E3. This will give rise to one type, one set of protein. So I can call it as protein 1. If there is a second combination of exons, E1, E3, E4, this will result in a second combination of protein. So this is also a true statement. So by observing, by carefully reading and analyzing these statements, we can come to the conclusion that the first two statements are true, the third one is false, and the fourth one is true. Correct answer in this case is D. Such elaborative type of questions are expected in the forthcoming NEET examination. So be prepared for such type of questions.